The main challenge for doing this type of work is that you need to have systems that allow you to test whether your method is actually working. So you need to find a biological system that other people have characterized well so that you can compare your results to what they have found. And so we've picked for the study two specific biological systems. One is a particularly promoter region in the mouse genome for the IGF-BP1 gene. This promoter region has been very well characterized, and there's a particular transcription factor called FOXO1 that selectively and specifically binds to this promoter region.
FOXO1 it has a DNA binding domain, so this is a domain of the protein that enables it to bind to DNA that has a very distinctive structure that looks a lot like that of the linker histone. Now, linker histone binds to the outside of the chromatin, and it compresses chromatin, so it doesn't allow transcription factors, other regulatory proteins to get in and activate a gene at gene regulatory sequences. What proteins like FOXO1 are able to do, based on studies from my lab and other labs, it can use this linker histone-like DNA binding domain to exchange with the linker histone, bind to its sites in the chromatin, and then open these regions up for binding of other transcription factors. And so as part of the process in discovering this, we needed to purify FOXO1 as a recombinant protein, and we also created nucleosome particles from DNA that corresponded to one of the promoters that FOXO1 binds to. This is the IGF-BP1 promoter, and these nucleosomes, so it's the DNA plus histones that are also purified, and these are assembled using a variety of techniques into purified nucleosomes. And then what we can do is bind FOXO1 to the nucleosomes, capture this on a solid surface, for example, gold surface slides, and then be able to apply the mass spectrometry protocol to be able to identify FOXO1, histones that have actually been captured on the surface. And this has been done very successfully as an initial part of this project.
And so we have both mutated forms of this promoter where the protein no longer binds, and the wild-type forms of this promoter region where the FOXO1 protein binds very efficiently. And so we can use this type of sequence as a control experiment to sort of see, do we find the FOXO1 protein bound to this DNA with our approach in the wild-type sequence, but not in the mutated form. The second system then that we use is a little more complex, and that is in the yeast genome.
My expertise is in yeast genetics, and one of the initial projects of the Wisconsin CEGS group is to bring this new technology to the yeast system.
I like yeast because they're simple. So, I mean, yeast is an organism that most people think of as being used for bread making or even beer making. But in the lab, they have a very small genome. They've been studied for a really long time. Nobody feels bad killing them. And the yeast as a research organism really has a lot of in-depth understanding. And so we can ask much more finely tuned questions because we have a lot of background already about which genes are involved with specific pathways, which proteins are in the right places at the right times.
Although yeast look nothing like humans, their cellular biochemistry and genetics is very similar to humans. And so what that means is that yeast is a very simple system that we can work in very quickly and quickly generate results that are transferable to other systems, including human biology. And so this is really what's known as basic research. We're using a model organism to understand fundamental basic parts of how the cells work, and this is able to generate information that we can use directly in other organisms, including humans.
Yeast normally can use multiple different carbon sources, and there's a crucial switch that occurs when carbon sources are changed. So when the yeast is grown with a different sugar. And one of the regions that mediates the switch is located between the GAL1 and the GAL10 gene in the yeast genome. And this particular region has a repressive effect. So it represses the transcription of the neighboring genes when it's grown under glucose conditions. And once you switch to galactose as a sugar source, these two genes get activated, changing dramatically what types of proteins bind to this specific region. And so this is the second model system that we use, where it's no longer just one protein binding, but multiple proteins binding, but it has still been extensively studied in the past, and so we can compare how well our methodology identifies these proteins that other people have reported as playing a role in this switch.
My current focus at the moment has been to really work on developing new methodologies to process these complex yeast cell lysates, because yeast is our model system, and figure out how, basically where the DNA protein complexes that are cross-linked to each other are going when we break open the cells and we have this complex soup, this mixture of all different kinds of stuff that only a very, very small portion of which we actually care about. What do we do to that sample to get the stuff that we care about out, and how do we treat the sample, and most importantly, doing this in a way that's friendly to proteomics analysis with mass spectrometry, which is really sort of the novel aspect of this project in a lot of ways.